and welcome to this masterclass series. My name is Alex Trotter. I'm a development scientist in translational development here at Oxford Nanopore, and I'm going to start you off on this journey through using the no-miss workflow for microbial isolate sequencing before my colleague Chris takes over for the second part. Now, this is part of our sample to answer series. There's a lot of great talks here, so please do go and check them out if there are any of them specifically relevant to you. But this one is a one-off special on the no-miss workflow. Now, this is a full end-to-end workflow, nanopore-only microbial isolate sequencing solution, and we're going to talk about how we can use that to sequence our isolates. Now, hopefully what we'll learn today is we'll learn how this workflow is going to enable you to do whole genome characterization of either your bacterial or your fungal isolates. We're going to walk through every step, okay? We're going to go from your sample preparation. We're going to talk about how we're going to do our extractions. We're going to talk about what library prep we're going to use. We're going to talk about how we're going to sequence it. And then once we're sequencing, Chris is going to take over and talk you through using the Epitome platform to do your data analysis. And then we'll also look at a load of helpful resources. There'll be links at the end for these, for when you go in to do this yourself and you're trying to look back and think, right, what extra do I need to know? So let's get started with why we would want to use nanopore technology for this. Okay, so let's talk about the microbiology and infectious disease applications we can do with Oxford Nanopore technology. Now, the one we're going to focus on here, obviously, is bacterial isolate whole genome sequencing for no-miss. What we can do with this, we can be doing outbreak investigations. We can be looking for antimicrobial resistance, find those AMR genes. And we can also be doing molecular epidemiology. We could be doing pandemic preparedness with this. But there are other applications we could be doing within this space too. Full-length 16S. Using long nanopore reads, we can get the entire 16S gene. We can get, then, for better taxonomic resolution in that, we could be looking at polymicrobial defect detection. We can do whole microbial community profiling. We've also got some great end-to-end options out there for viral amplicons. We offer workflows for influenza A and B, SARS-CoV-2, and these can provide some really critical public health information. Also, we could be doing metagenomics. Long nanopore reads can generate metagenomic assemblies to characterize complex communities. We can do agnostic pathogen detection if we don't know what we're looking for. And we can do rapid AMR profiling as well. Now, let's talk about the advantages of nanopore sequencing here. Some of the great features for us here, the platform is accessible, right? We're talking really small machines. If you've seen a MinION, it fits on the palm of your hand, right? So these platforms don't take much space. They also don't have high upfront costs. So getting started is really easy. Anyone can just get stuck into nanopore sequencing if they're interested. Now, we're also doing real-time data generation here, and that means we can perform rapid and dynamic turnaround times to get those important answers sooner, where it really matters for certain applications, sort of how fast we can get this out. It means we can be flexible, right? You don't need to batch all your samples together in huge runs. You don't need to think, oh, right, yeah, the sequencing run is on next Thursday, so I'll just wait for that. No, when you're ready, you can go. You can use however much you need, and then if you don't need to run a flow cell to completion, you can stop, you can take it off, you can wash it, recalibrate it, and use it to store it for later. And this can be really essential for some applications. Let's think, public health interventions, outbreak tracking. You need those results back today. You can do that. Now, we also have the ability to generate these really long nanopore reads, and I'm going to go into a little more detail on why this is really useful in the next slide. So, one problem with short read technology for this application especially is genome assemblies can end up fragmented, they can be incomplete. Let's have a look at this image on the left here. This assembly is just lacking clarity. There's no understanding coming from this of what's in here, where things are, and how they might interact with each other. Nothing connects. Whereas if we look at the assembly on the right, using accurate nanopore-only long reads, we get a complete circularized genome. We can resolve plasmids out of the chromosome, and we can locate where those AMR genes are. We can understand how they interact with the rest of the genome and take a real dive into what's driving antimicrobial resistance here. Now, here we have an overview of the nanopore-only microbial isolate sequencing solution, no-miss for short. And we're going to go over that together, okay? Start to finish, extraction to analysis, sample to result. Step one, get your isolates. But from then on, we're going to talk about which extraction is going to be right for you. We have a few options here for extractions. Not every extraction will be able to do every organism. Some are quite difficult if you're taking an enzymatic approach, and we'll run through that. But then after that, after you've done your extraction, it's all coming back together. We're going to talk about sequencing using the rapid barcoding kit for our library preparation. For this, we're going to recommend putting 24 samples on a run, but that's flexible. You can do fewer if you need to do fewer. Or alternatively, you could do more. If you know what organism you're working with, let's say you know you've got a smaller than average genome size, you know that the same amount of data is going to go further for you. So you can up that. Now, after that sequencing, we're going to then go, and Chris is going to walk you through how we're going to analyze that data using epitome. Now, potentially the most important thing we're going to talk about is extractions. We've already had a great talk in this series by Vanya about extractions, so please check that out if you haven't done already. For this application here, we have no PCR. We are sequencing native DNA. So what you extract out is what we're going to sequence. That's what's going in. So it's really important that that extraction is free of any contaminants. It's going to be free of sequencing inhibitors, and it's going to be free of proteins and RNA. The methods here in this protocol have been optimized for sequencing yield, so we really recommend following them as closely as you can to get those best results. So we have 4 extraction protocols available here, and this is going to depend on both what organism you're using, but also what you're looking to get out of it. So on the right-hand side here, we have a mechanical lysis option. It's going to use vortex bead beating. Now, this is the universal protocol I would recommend to most people. It's simple, it's quick. You don't need to worry about what organism you're trying to extract. This is a real one-size-fits-all approach. Now, automated prep is written here because the extraction kit that we recommend for DNA purification after you've done this lysis is an automated mag bead extraction. But that's just to keep this as the most simple, straightforward, and hands-off method in here. But don't let that put you off if you don't have an automated extraction robot. You can do a manual extraction, a manual cleanup from the other methods after you've done this vortex bead beating lysis. Now, this will give you potentially shorter reads. If you're bead beating your extractions, we're looking at an N50 of, say, on average, 4 to 7 KB reads. There will be longer reads in there as well, but that average is a bit lower than if we were doing an enzymatic extraction. Now, if you're looking for the best quality extractions, you're looking for the best genomes, the longest read lengths, an enzymatic lysis approach is probably what's going to be for you, right? So these are a little more time consuming, a little more complex, but they will give you more highly contiguous assemblies. We will be getting slightly longer reads out of them. So if you choose to go enzymatic, we're going to have to pick which method's going to be best for you. Now, we have tried to keep it mostly universal. We have a universal bacterial lysis method here that's going to use lysozyme, and this will work for your gram-negatives, most of your gram-positives. You might be thinking, hang on, I've got a gram-positive in Staph aureus. That doesn't lyse with lysozyme. Don't worry, inside the protocol there'll be recommendations of substitutions of enzymes and buffers if you're doing a tough organism like Staphylococci. It's all in there in the protocol. Alternatively, for some really tough to lyse organisms, we have some other extraction methods listed here as well. We have a hard to lyse method that's been designed for Mycobacterium tuberculosis, notoriously difficult to lyse enzymatically. And you can walk through that for that organism. But the nice thing about this as well is it can be used as a fallback if, let's say, you have a tough organism that won't lyse through the normal bacterial method. You can try putting it through this tough to lyse one. We found in development a couple of organisms, Strep A, for example, tough to lyse, wasn't giving us good yields when we put it through the generic bacterial extraction. But when we put it through the tuberculosis hard to lyse one, we were getting good extraction yields. So that's there for you if you need it. And also we've got an enzymatic extraction for fungal cells. But all of these could go altogether through the automated one. As I said, that will do your bacteria, your hard to lyse, and your fungi. It's just picking which method is going to be right for you. And after that, everything comes together for the rest of the protocol. So now we're going to talk about library prep. And the library prep kit we're going to recommend for this is the Rapid Barcoding Kit, RBK. So RBK is a PCR-free library prep. It's great for when turnaround time really matters to you. As a library prep, the incubation of the actual library prep reaction is about 5 minutes. In total, the preparation should probably take you less than an hour. Now, these kits are sold to do up to 24 or 96 samples. That's how many barcodes will be in each kit you get. We've spoken before about no miss. We're going to recommend to 24, but as we've said, you can flex that. If you're needing to put more than 24 isolates on a run, you might want to get the 96 kit to use that. Now, RBK uses a transposase to cleave the DNA, and then it's going to add in the barcodes, it's going to add in the sequencing adapters. This does fragment your DNA a bit, but we're still getting those long reads. As you said, enzymatic lysis, we're looking, most reads will probably be about 7 to 15 kb, but there'll be reads in there that are well over 60 kb as well. Okay, so now let's move on to actually doing the sequencing. So when it comes to sequencing, other masterclass sessions will have given you way more information on all the types of flow cells, so please do refer back to those if you're interested in the full range. For NOMIS, I'm just going to talk about MinION flow cells. Now, MinION flow cells can be used for loads of different applications. They're really flexible, but the yield fits nicely for what we're trying to do with NOMIS here, for that recommendation of 24 isolates to get us multiple isolates, high depth on one flow cell, 24 in 48 to 72 hours of sequencing. That should be giving us at least 50x coverage per sample, which will be good for most applications. But your applications might need to tinker around to get more or less, depending on what you need. Now, it's also going to be the really flexible thing about this is that we can run less, but we can also wash and reuse them. Let's take an example of a public health application, outbreak tracking. Maybe we need to run 3 or 4 isolates really quickly. We need to know, are these linked? Yes or no? Do I need to make a change? I need to know today. We can do that. We can run a few samples in a few hours, then wash that flow cell, recalibrate it, store it in the fridge, and use it later with different barcodes on a new experiment. That way, we're both keeping the turnaround time down, but we're also keeping the cost per sample down as well. We don't have to burn an entire flow cell, an entire sequencing run on one small experiment. And that's really useful because you might not want to do 24 at once. Maybe what's best for you is doing 6 isolates 4 times, still on one flow cell. Now, different flow cells pair with different devices. So let's talk about the MinION. It's simple, it's portable, it can take one flow cell at a time. It's ideal for this application and completely flexible. You can move it around the lab. You can take it out of the lab if you want. It really is just that easy to use and take about. Alternatively, something else that will take a MinION flow cell is the GridION. This is what I have in the lab. The GridION can take up to 5 MinION flow cells at a time, and they will run independently. So you can have multiple runs, you can have multiple people, or you can have multiple projects all going on the same time. This is a benchtop machine. It's got onboard computing, so it's quite a neat way to keep everything together, everything really organized, so long as you don't need to move it about. Now, once our flow cell has been put on the sequencing device we've chosen, it's time to get ready, it's time to go, it's time to sequence. Now, sequencing is going to be controlled by our onboard software, Minnow. We can see a screen from Minnow on the top left here. You can watch its performance go in real time with all the colors, which can be quite mesmerizing, but it's very useful to us. We can see while the run is going exactly how it's performing. We can see certain metrics to it. We can see our read lengths. We can see which samples are sequencing to what degree. We can see how it's going. We don't have to wait until the end of the run, until sequencing's finished to know what happened. We can look at it in real time. Now, that software is also going to do the base calling for you. Again, that's in real time. So that data is going to be ready as soon as the run's finished, or even before. If you want to take, say, a snapshot of the first few hours, you want to get some insights, some sequencing results straight away, we can do that. We can take that off. We can look at it while the rest of the run is still going. And that data output is then going to go into our analysis software, Epitome, and for NOMIS, we're going to be using the bacterial genomes workflow, which Chris is now going to take you a deep dive into, walk you through how to use it using a salmonella isolate as an example. So thank you for listening. And Chris, over to you.
Hi, my name's Chris Older. I'm a bioinformatics workflow developer within the Epitome team here at Oxford Nanopore. Alex has taken you through the extraction, library prep, and sequencing portion of the NOMIS workflow, and I'm here to briefly go over the data analysis side. We've released a new version of the Dorado base caller, version 4.3, available within Minnow. This version has base calling models trained with native bacterial DNA, which has helped fix errors associated with methylated sites found in previous versions. Information on these methylated sites come at no additional sample prep for the user. With this new methylation-aware base calling model, combined with the R10 flow cells in Kit 14, we have improved the overall quality of our bacterial genome reconstruction, enabling reference quality genome assembly without the need for short read sequencing and hybrid assembly. One of our key aims in the EpiTME team is to enable bioinformatics analysis for everyone. To help with this, we have developed the EpiTME desktop application for analyzing your nanopore data. It's available on Mac, Windows, and Linux systems. It has an easy-to-use interface with a number of pre-configured workflows ranging from human genetics to metagenomics. These workflows can be run locally or now in the cloud, with some workflows allowing analysis in real time. Each workflow produces interactive reports and standard output files. For more advanced users or those comfortable with it, the workflows available within the app can also be run on the command line and are hosted on our GitHub. If you want more information on EpiTME, please check out the masterclass session entitled How to Analyze Your Data in EpiTME. For the NoMIS workflow, we'll be focusing on WF bacterial genomes, which is EpiTME's workflow for assembly, variant calling, and annotation of bacterial genomes. Here we have an overview of the processes within WF bacterial genomes. This includes the assembly, which can either be performed through a de novo or reference-based method, as well as annotation. The workflow also includes a module entitled Isolates, which provides further analysis on your research samples. To run WF bacterial genomes locally, you'll need a computer with a minimum of 16 gigabytes of RAM or 4 CPUs. However, we recommend one with 32 gigabytes of RAM and 16 CPUs for optimal performance. For sample yield, we recommend a minimum of 50x coverage to ensure high-quality assemblies and confidence in your analysis results. Different workflows within the EpiTME platform will have different sample and compute requirements, so please refer to the documentation of each workflow for specific recommendations. Each workflow also provides some demo data to use as a test run. We recommend running this to ensure the workflow will execute properly on your machine. It's also a good opportunity to allow the workflow to download additional containerized packages needed to run the analysis. To get started assembling and annotating the data with the workflow, we simply need to provide the FASTQ or BAM files of your research samples. You can also provide a sample sheet, which can be used to dictate the sample names for the workflow outputs and report. If running in reference-based mode, you'll also need to provide a reference faster. For each sample, the workflow will produce a draft genome assembly, a genome's annotation file, and a variant calling file if run in reference-based mode. The results will also be available for each sample within the interactive report. The workflow can also provide additional analysis via its isolates mode, which can be activated within the app for a simple checkbox. This includes multi-locus sequence typing, which helps characterize your organism using a species or genus-specific typing scheme. There are typing schemes available for most common bacteria. The workflow will automatically determine the most appropriate scheme to use for your research sample. Next, we have antimicrobial resistance analysis. This will detect the presence of acquired resistance genes associated with AMR in your research samples. For a select number of species, such as Enterococcus faecium or Mycobacterium tuberculosis, the workflow can also detect AMR-mediated mutations. Finally, we have serotyping. This is available for Salmonella and will provide information on predicted serotype and antigenic profile. Some of the analyses in the isolates modules are species-specific, but prior knowledge on the sample is not necessary. The workflow will detect the species from the multi-locus sequence typing analysis and apply any species-specific analysis if applicable. In isolates mode, the workflow will also produce a report for each sample on the run, which highlights key information about your research sample. And with that, we've reached the end of the NoMIS workflow. To end this masterclass, we'll quickly go over some extra resources you may find useful. First up, on the left-hand side, we have a link and QR code to learn more about other microbial research you can perform using Oxford Nanopore technologies. In the middle, we have a link and QR code to a workflow within our resource center, which introduces the use of NOMIS to bacterial isolate sequencing. And finally, on the right-hand side, we have a link and QR code to access all the protocols available through the Nanopore community page. I'd like to thank Alex for helping me showcase the NOMIS end-to-end workflow, and thank you for attending this masterclass.